Okay, do believe I should be. Yeah, I am recording. Okay, so where we left off, I've been talking about this guy named Carl Linnaeus, the Swedish botanist with an ego that would not fit through barn doors, who, for both religious reasons and for very practical reasons, was trying to come up with a way of standardizing names of species. And on Friday, I talked about his practice of using so called binomial names, where biological species should each get a standardized name that would be in Latin, or at least spellable and pronounceable in Latin, that would consist of two parts: one word for the genus, followed by one word for the species. I'm being a little bit loose with the definition right now. We're going to get a better one coming up, but species you can think of for the moment as just being basic type of living thing, and a group of species that are all similar in important ways would be a genus. So all three of these are roses, so they go in the genus Rosa. But each one is a different species of rose. So we call them Rosa canina, Rosa gallica, and Rosa grandiflora. And again, it works a little bit like your names work, except the equivalent of the last name goes first, right? Just my name is Wagoner, which tells you what, group of people I'm a part of, what family I'm a part of, and then Ben to specify which one of the family I am. This works the same way, except we reverse the order and we put the general name first. And again, we still use names like this for all species just by international agreement. There's no law that says you have to, but this is just what we've all decided on as a convention. Latin remains the language that we use because Latin is not the language of any major world power. So people from all countries are kind of starting from the same starting point when they have to use these names. When we write them, by the way, you always include the genus and species names together. You can't use only the species name. I'm not going to bust your chops on this as far as grading goes, but the names are italicized, you capitalize the genus, and you never capitalize the species name. So all of these names for this fish you can use if you want to. If you're Russian, you're still free to call it a Sinye Jabrny Solnichnik. But in scientific context, we'd refer to this as Lipomus macrochirus. And of course, not every biologist is going to know that name, but at least we'd be able to figure out that's where we would look it up. That's where, if you want to think of it in this terms, this is what Wikipedia would call it. You may already know some of these. You might know that our own species is Homo sapiens. Homo is the genus, and it's just the Latin word for a human person. Sapiens is Latin for wise. So Homo sapiens means wise man. And Homo is the genus, and sapiens tells you what species we're talking about. Now, exactly what is a species? Again, this is something we'll get to in a little bit more detail later. For right now, we can say that Linnaeus defined species by distinctive physical features that they have. So this happens to be another tasty fish that puts up a great fight, that's fun to catch in lakes of Arkansas. It's stocked here, and it's called Micropterus salmoides. Micropterus is the genus, Salmoides is the species. And what defines it, at least the way Linnaeus would have done it, was that all Micropterus salmoides have a distinctive physical features that none others have. So Let me see if I can. There's the laser pointer. Right there is the back edge of the eye. This thing right here, shaped kind of like a teardrop, that's the upper jawbone of the fish. And the rear end, the back edge of that upper jaw, right where I've got the pointer, is behind the rear edge of the eye. That's what make all fish that look like this Micropterus salmoides, aka the largemouth bass, and not Micropterus dolomieu, the smallmouth bass, or Micropterus punctulatus, the spotted bass, or Micropterus cusei, the Alabama bass, or the striped bass, which is not even in genus Micropterus, and I'm suddenly drawing a blank on what it is. But the point is, a species for Linnaeus would be all organisms that have at least one distinctive physical feature that they share in common, and that they don't share with any members that are not of the species. The problem is that's not really a great way to do it because it turns out that individuals in many species vary a lot. You can see this in our class. We're all Homo sapiens. We've got people of different heights, different builds, different hair color, different hair texture, different skin color, different eye color, different proportions. We're a variable species. We vary quite a lot. Here, let me admit. Um, Here's another example. These are all considered one species. This is a land snail from Cuba called Polymita picta. And you can see that each shell is different from the others, in proportions, in size, in fine details of the shape, in background color, in bands, in number of bands, in color of the bands. And maybe you could try to divide Polymita picta into separate species, but it's not immediately obvious where you would draw the lines. Which of these variable features are the most important for dividing this thing up? Well, you know, that's kind of a hard problem. Species don't come with nice, neat name tags that would make everything easy. So the old way of defining species was called the morphological species concept. I'll never test you on the meanings of these words, but it might help you learn the vocabulary. Morpho is the Greek word for the shape or the form of something. So the morphological species concept defines a species by its physical features or its traits. This is still what we use practically in a lot of cases. If you buy a field guide to North American birds and you go out bird watching, you'll have to identify birds from pictures using physical features. So you'll pretty much use the morphological species concept there. The problem is that, as you've seen, it can be ambiguous, especially because a lot of species, heck, all species that I can think of, have some degree of variation. Just like we have variation in all of our physical traits, every species has that to some extent. So we've largely replaced this by what we call the biological species concept, where we define a species by its members' potential to interbreed. Everybody in this class is in the species Homo sapiens. We all look different from each other, yet I'm not actually recommending this, but theoretically, um, everybody in the class could, in theory, not in reality, pair up with each other and have kids. Maybe a few might be infertile or something like that, but most of us could have kids with most of the rest of us if it were to come to that. Any two humans of opposite sexes that are otherwise healthy can get together and have kids. And in fact, that's been going on all through human history and prehistory. And so it is with all other species. We wouldn't necessarily define largemouth bass as all bass that have an upper jaw that extends past the posterior margin of the eye. We define it as a set of populations of bass that, in principle, can all interbreed with each other and make fertile babies. And a species is said to be reproductively isolated from all others. Members of one species can't usually produce fertile and healthy offspring by breeding with other species. Even that is an oversimplification because we now know that a lot of entities that we thought of as separate species can interbreed and make fertile offspring, even if they usually don't. So, for example, polar bears and grizzly bears are usually thought of as separate species. We now know that, in fact, they can interbreed, and there are, in fact, populations of bears in Alaska known as pisleys that are hybrids between polar bears and grizzly bears. This actually can get a little bit fuzzy, and this actually leads into about a week's worth of lectures that I give in one of my upper division classes. So even the biological species concept can be a little bit fuzzy. Getting into that is going to take more time than we've got. So this is one case where I'm just going to wave my arms and say, as far as we're concerned, we'll go with the biological species concept. If you get into upper division classes or read more on the subject, you might find out that's not a perfect concept either, but this is what we'll go with for the purposes of 1400. It'll do for what we need to accomplish in this class. Under the biological species concept, we sometimes find species that can't be told apart by looking. Um, these are considered two separate species. That's a Carolina chickadee on the left. You get these around here. In fact, this time of year, if you were to set up a bird feeder and fill it with lots of bird seed, you'd probably attract them if you put it in the right place. And it's called the chickadee, by the way, because that's the sound that it makes. They're cute little birds that go chickadee, except about 3 octaves higher than I can make my voice go. Anyway, on the left, that's the Carolina chickadee, Posile carolinensis. On the right, the black-capped chickadee is Posile atricapillus, and you can't tell them apart by looking. Physically, they are practically identical. We call them separate species because they don't interbreed. They don't make babies. The black-capped chickadee, by the way, lives farther north of us. It would live roughly north of St. Louis, Missouri. So we only get the Carolina chickadee. But in places where you get both, they don't interbreed. A major reason why they don't interbreed is that birds recognize mates, many of them, many birds recognize mates by song, and males of one species sing songs that females of the other species simply don't recognize or are not attracted to. So in practice, they don't interbreed, even though they look identical, and we would call this a case of cryptic species. So you can have two biological species that are perfectly well defined and that don't interbreed, but that you can't tell apart by looking, and we would say those are cryptic. The second big thing that Linnaeus did, aside from binomial nomenclature, was coming up with what amounts to a filing system for all of these species. Coming up with what we would now, in modern terms, call a database, a way of organizing all of these data. And we've already seen that species that are similar go into the same genus. The lower left, that is Lipomus macrochirus, that's the bluegill. Up at the top, Lipomus gibbosus is the pumpkin seed. The green sunfish is Lipomus cyanellus, and I think there's about 9 or 10 more. I'm not sure of the exact count. I mean, there's the orange ear sunfish and the pygmy sunfish and the warmouth sunfish. There's more that I don't recall. I'm not a fishologist, but overall, there's I think about 10 or a dozen species of fish in North America that all belong in the genus Lepomis. So similar species go into the same genus. Genera that are similar go into a larger group called a family. So down there at the lower left, those are four out of the dozen or so members of the genus Lepomis, commonly called sunfish. At the top left, those are two members of the genus Pomoxis. We commonly call these crappies. Those used to be stocked in Lake Conway. My neighbor used to have a boat and go out and usually not catch anything, but he would go fishing for crappie in Lake Conway. So if you fish, you might know crappies. At the lower right, genus Micropterus is the freshwater black bass, and that would include not the striped bass that you'd catch up at Greer's Ferry. That's in a different genus, but largemouth bass, smallmouth bass, Alabama bass, a couple of other bass species uh, on the eastern seaboard. So we have three genera of fish. You can see that they're all similar to each other. And the three genera all go into a larger grouping called a family. And the family in this case happens to be called the Centrarchidae. For the record, you do not have to memorize how to classify fish. I mean, you might if you ever take an advanced course. We do teach courses in fish, but for our purposes here, you don't need to memorize the name Centrarchidae or anything like that. I'm giving this as an example to help you understand the more general concept of what we call the taxonomic hierarchy. But you do not need to remember that Pomoxis is the genus that crappies belong to, or that largemouth bass are Micropterus salmoides, or anything like that. Focus on the general principle here. And then if you have multiple families of similar organisms, they go into a larger group called an order. If you have several orders that are similar, they go into a larger group called a class. If you have multiple classes that are similar, they go into an even bigger group called a phylum, Phylum. I'll show you that in a minute. Phyla that are similar go into an even bigger group called a kingdom, and kingdoms that are similar go into the most inclusive group of all, a group called a domain. So don't worry about remembering the names that I'm going to give you, but here's how the groupings work. If we have this fish, it belongs in one of three domains of life. All life that we know can be put into one of the three domains, and this specific domain is the domain eukarya, which consists of all living things whose cells have a DNA containing sac in the center called a nucleus. I'll talk more about that in just a minute. So this is in the domain eukarya because all of its cells contain these little bags of DNA in the middle called nuclei. The eukarya can be divided up into multiple kingdoms. The best way to do that, people still discuss, but the kingdom that we'd put this fish in is traditionally called the kingdom Animalia. We'll be covering several of the kingdoms of eukaryotes, and this one is in the kingdom Animalia. Those are members of the eukarya that are made up of multiple cells and that have not just many cells, but many different types of cells in their body and hold their cells together with a meshwork of proteins called the extracellular matrix. We'll talk more about that later. But the kingdom Animalia would include everything from corals and jellyfish and sponges, all the way to fish and birds and mammals and bugs and spiders and quite a lot of other things. The kingdom Animalia you can divide up into roughly 25 phyla. The plural is Phyla. And within the kingdom Animalia, this fish goes in a phylum called Chordata. Chordata are those animals that have a nerve cord running down the backside. It's the spinal cord, and a support structure supporting that nerve cord called the notochord. In fish, it's mostly replaced by the vertebrae, and it is in you, but you had a notochord when you were a tiny embryo, as did this fish. And there's a few other features that make this fish a chordate that put it in the phylum Chordata. You traditionally can divide the Chordata up into 7 classes, and this fish goes in the class Actinopterygii, which is kind of a mouthful to say. It means fish that have fins supported with rays. If you look at those fins, you can see, that the fins are very thin, very flexible, but what holds them up are thickened structures, which in some cases may be very flexible and in others may be quite sharp and bony. You can see the spine-like fin supports up here in this part of the fin. And so all chordates that have fins that are supported with these thickened structures called rays go in the class Actinopterygii. You can divide Actinopterygii into multiple orders, and this one happens to be in the order Perciformes, fish that are shaped like perch, that, for example, have spiky rays in the front and flexible rays in the back of the dorsal fin. You can divide the Perciformes into multiple families, and one of these is the family Centrarchidae. The Centrarchidae contains several genera. This one is genus Lipomus. That's the American sunfishes. And Lipomus contains 10 or a dozen species. This one is the species Lipomus macrochirus. Okay, and again, don't worry about memorizing Actinopterygii or Centrarchidae or anything like that. What I want you to get the sense of is that species can get clustered into more inclusive groupings, and then those getting clustered into even bigger groupings, and those getting clustered into even bigger groupings, and so on until we get to the largest groupings of all. You could think of each one of these groupings as just being a file folder on your desktop. Um, I don't know how you organize the desktop on your computer, but I might have a folder on my desktop that says Biology 1400. And within that, I'd have subfolders for each year. And within that, I might have subfolders for lectures, labs, etc. And within that, I might have more subfolders. So if you spent a lot of time organizing your files on a computer, you're accustomed to the idea of folders within folders in this hierarchy. And species are grouped together in much the same way. Each one of these groups is just a folder, and each one of these folders goes in a higher level folder and contains lower level folders. Hopefully that makes a certain degree of sense. So from here, that's all I'm going to say about the hows of how we do biological classification. It's probably not the most interesting thing you'll ever learn. I want to move on to going through um, the domains and kingdoms of life, and I'm going to start with those 3 domains. All life that we know goes into one of these 3. The domain eukarya consists of all living things where the cells contain nuclei, and the nucleus of a cell is a flexible sac in which the genetic material is stored, the DNA is stored. About midway through the course, we're going to get into more detail about what's in nuclei and what that DNA stuff really is and what it looks like. We'll get to that a little bit later. Eukaryotic cells are also compartmentalized. They contain a lot of specialized compartments that carry out different functions, that play different roles in the overall metabolism of the cell. I like to compare eukaryotic cells to things like the Lewis Science Center, the building where we normally have lecture, because that building contains a lot of rooms and it contains different types of rooms that are specialized for different things. We have big lecture rooms, we have labs, we have a conference room, we have faculty offices, we have department offices, we have storage closets, we have rooms for the HVAC, the heating and air conditioning. We have lots of rooms in that building, and each one of those rooms is built to handle a specific thing that we need to do. So Lewis is highly compartmentalized, and we have different compartments for different functions. Eukaryote cells are like that. Eukaryote cells can be divided into many compartments, and each type of compartment does a specific thing and contributes in a specific way to the cell's overall life. So eukaryote cells contain nuclei, and they contain these compartments that we call organelles, analogous in some ways to the organs of your body, where you have, a heart and a liver and lungs and a pancreas and kidneys, each one of which does something different to sustain your life. It's analogous in some ways to the organelles of eukaryotes. Cells that don't have a nucleus, they still have DNA, but the DNA just kind of floats in there. It's not compartmentalized off in a sac. Cells that don't have a nucleus and don't have organelles are said to be prokaryotes. Eukaryotes are the ones with a nucleus, prokaryotes don't. And we used to consider the prokaryotes a single domain, but beginning in the 70s, a guy named Carl Woese at University of Illinois showed that there's actually two types of prokaryotes that are fundamentally different from each other in a lot of ways. And we now call these the archaea and the bacteria. The differences between archaea and bacteria are another one of those things where I'm just going to kind of wave my arms and tell you to go look it up on Wikipedia if you really want to know. There's some very fundamental differences in the ways that archaea and bacteria are organized, the types of macromolecules that they have. For our purposes, we don't need to get into that. You're perfectly capable of understanding it, but it would take more time than we've got. And I'd need to go into some chemistry background that not everybody has. And again, we've got a huge amount of material to cover. So this is another one of those cases where I'm just going to kind of arm wave at you. You're fully capable of understanding the difference, but again, we just haven't got the time. So I'll vaguely suggest you can look this up if you ever want to. Archaea and bacteria are both prokaryotes. You can't easily tell them apart just by looking at them under the microscope. They've got very profound differences in their biochemistry, in their metabolism. We're not really going to get into what those are. The only thing I usually teach about archaea is that many of them live in environments that are extreme. They live in environments that are too hot or too cold or too salty or too acid or too alkaline or otherwise too extreme for anything else to live in. Now, many archaeans are not extremophiles, and some bacteria are extremophiles. So this is not a good dividing line. Um, but the fact that many archaea are extremophiles is really the only thing I'm really going to cover about archaea as such. So in an upcoming lab, it's lab two, but I think it's you're not doing it for another couple of weeks. You are going to look at eukaryotic cells under a microscope. One of our lab exercises, you're going to have to scrape out some of your cheek cells. And when you scrape the inside of your cheek and smear the result on a little piece of glass and add a drop of blue dye, you'll see this. These are cheek cells. And in the center of each one, there's a little blob that stains very strongly blue. That's the nucleus. Each one of these blobs is the nucleus of one of your cheek cells. You are a eukaryote. Um, in your cheek cells, you can't clearly see the organelles, although they are there, but you're also going to look at a piece of plant leaf, a water plant with very thin leaves in the genus Elodea. And in each cell, you'll see a whole bunch of green little dots, look kind of like green jelly donuts, floating around inside the cell. And those green dots happen to be organelles. They are a type of organelle called a chloroplast. Um, what they do is they are where the actual work of absorbing light energy and transforming it into a chemical type of energy that can be stored. They're where that takes place. They are where photosynthesis takes place. And again, we'll get that again a little bit later. I just picked this because this is a case of organelles that you can clearly see. A lot of eukaryotic cell complexity, however, you can't pick out with the light microscope. And to see it, we have to use a much more expensive gizmo called a transmission electron microscope. We don't have one of these at UCA, although they have got one at UAMS and at UALR. What you're looking at here is a slice through a plant cell um, that has been specially stained and visualized using a transmission electron microscope. And this can magnify far, far more than any light microscope can. So what you're looking at here is in the middle, there's this grainy blob. That's the nucleus. Inside the grainy blob, there's an almost completely dark blob called a nucleolus. We'll talk later about what that does. The bigger grainy thing is the nucleus. And on the inside, you can see, okay, these gray dashes right there and there and there and there. Those are organelles. These open spaces are organelles. There's various others that are embedded in here. We don't really need to worry about what those are at this stage. We will cover that later. We'll come back to this. What I want you to see is that that cell looks compartmentalized. You have lots of different spaces in the cell, lots of different chambers, lots more complexity in how that cell is put together. If you take a picture of a bacterial cell with the same microscope, you can see these are just a lot less complex. They're not divided up into compartments. The DNA is kind of this light gray blob in the middle, but it's not partitioned off in a separate room of its own. It's not in a nucleus. The other structures that bacteria need to live are all just floating around on the inside, so you don't really see a nucleus or organelles. Um, I like to think of bacteria and archaea as being more like that indoor practice facility the football team has, where it's basically one big room where everything happens in that one room. As opposed to a eukaryote, which would be more like a typical academic building, which would have lots of rooms for different functions. So cells that don't have all those compartments that you saw are said to be prokaryotes. And the two prokaryote domains are bacteria and archaea. Up there at the top, these are also taken through a transmission electron microscope. Up top, you see several archaea cells that have been sliced through. At the bottom, those are bacterial cells. And even with this microscope, there really aren't any differences that you can see. The differences get down into the biochemistry level. And as I've said, we're just not going to cover them here. You could start by looking this up on Wikipedia if you really want to know more. Typical archaea and typical bacteria are extremely small. Typical sizes would be between 1 and 5 millionths of a meter. In case anybody did not know, we use the metric system here in science in general. And I assume you know this. My son has gotten the metric system every year, so I assume you all had it in your classes. A meter is, I still think of it as being a little more than a yard. A millimeter is 1,000th of a meter. So a millimeter would be about the thickness of a standard paperclip wire. And a micrometer is 1 millionth of a meter, or 1,000th of a millimeter. And micrometers are commonly called microns. So you'll see that thrown around. A micron is 1 millionth of a meter, and typical prokaryote cells would range from 1 to 5 microns in size. There are some that are smaller and there's some that are bigger, but that would be a typical range. If it helps, human hairs vary in size, but a typical human hair would be something like 50 microns. So you could take 50 bacterial cells and put them in line, and you'd have a line that's as long as the thickness of a typical human hair. Although, again, that varies depending on whose hair it is. There's not one single standardized hair thickness. The only thing I'm going to say about archaea is that many of them live in extreme environments. This is what you used to see flying into San Francisco International Airport. This is what you see at the south end of San Francisco Bay. These are man-made ponds. They're owned by the Cargill Salt Company. And what the company does is it will periodically dig a passageway and let water from San Francisco Bay, which is salty ocean water, flow into these ponds. And then they close each pond and they let each pond dry. And as each pond dries out, the ponds get saltier and saltier and saltier because the water evaporates and the salt is left behind. And as they get saltier and saltier, fewer and fewer living things can survive in them. And the saltiest ponds of all are these down here at the lower right, the ones that are starting to look definite reddish purple color. They look reddish purple because the only thing that can live in these ponds are archaea that are purple and are, as we say, halophilic. They can tolerate extremely high concentrations of salt. Just about anything else would die horribly in these ponds. If you were to drop a goldfish into one of these purple ponds, the poor thing would just shrivel like a raisin. I don't know, within seconds maybe, to be killed pretty fast. You certainly couldn't drink this water. It would play merry hell with your kidneys. But these ponds down here are purple because there is one species of archaean that happens to be purple when it grows in large quantities, that can live perfectly happy in water that is far saltier than anything in the oceans. So the purple ponds of the south end of San Francisco are purple because of extremophile archaea. They live in water that's extremely salty too salty for anything else. So yeah, you should never try to insult an Archean because they'll get salty. Eh, it's all right. You don't. I'm imagining you all laughing hysterically. There's an aerial shot of Grand Prismatic Spring in Yellowstone National Park, and just to give you an idea of scale, that right there is a boardwalk with people on it. So the whole thing is the size of a pretty large pond or small lake, but you would not want to swim in there. And unfortunately, several years ago, there was a guy walking his dog on this boardwalk, and the dog jumped into the water, and the man followed the dog, and they both died because that is a volcanic hot spring. The water in there is heated to 60 Celsius or so. I forget the exact temperature, but it's comparable to your coffee. You couldn't swim in it. Your skin would start peeling within seconds. You'd start suffering, third degree burns. What gives it its colors, though, is the fact that all of this yellow and orange mats right there, those are mats of enormous numbers of species of archaea that can live comfortably in water that is far too hot for anything else. You drop a fish in there and you're going to have fish stew very, very quickly. You swim in there, you'll get third degree burns and die in minutes. But the archaea that live in here can live perfectly well in this water that's nearly boiling. So we call those thermophilic, means heat loving archaea that can live under very high temperatures. And that's another type of extremophile, the archaea of volcanic hot springs. That's a river in southern Spain that flows through some rocks that have a lot of mineral deposits, and they make the water naturally acidic. We measure acidity on what we call the pH scale, and this water is pH 2, which means it's about as acid as lemon juice. And it's colorful. If you know Spanish, you know the Rio Tinto means colored river. That water is flowing orange because that water is not just very acid, it's rich in dissolved heavy metals, things like cadmium, mercury, arsenic. That stuff would be terribly poisonous to you if you were to drink the stuff. But there are archaeans that are called acidophilic that can live in conditions that are far too acid for anything else. So acidophilic archaeans can live in this river that is as acid as lemon juice and that's loaded with toxic heavy metals. Not a whole lot of other critters can live in there. A fish dropped in that river would die horribly. The archaeans can do just fine. And that's a third type of extremophile. There are even archaeans that live in places like this. That's about 5,000 feet down below the surface of the eastern Pacific. And what you're looking at that looks like black smoke, that's actually superheated water. It's heated by liquid rock below the surface. It's heated by magma, that's molten rock, and it comes shooting out of the bottom of the ocean at temperatures that can be higher than 100 degrees Celsius. That water only stays liquid because it's under pressure. That water would be boiling if you brought it to the surface. And it looks black because it's loaded with particles of iron and sulfur. So it's toxic, superheated water coming out into the ocean, coming into contact with water that's about just above freezing. And right in the middle of this crazy environment where superheated water contacts chilled water, there are extreme thermophile archaeans that can live at temperatures over 104 Celsius, over 220 degree Fahrenheit. So that's yet another type of extremophile. Only a few highly specialized organisms can survive in conditions like that. If you were to jump into that, I don't know whether you'd be burned to death or crushed to death first. They both happen to you at the same time. But these extreme thermophilic archaea do just fine in there. They would do perfectly well living in a pressure cooker. So much respect to the archaea for being some of the toughest organisms on the planet, able to survive under conditions where almost nothing else can. And we still don't know if there's life on other bodies in the solar system, but if there is, it's likelier to look like these Archeans than it is to look like the Klingons or the Kardashians or the Star Wars aliens in the Cantina or whatever whatever your favorite science fiction might be. Life like this just might be able to survive in places like some of the moons of Jupiter, or possibly on the surface of Mars. Um, we don't absolutely know. We won't be able to test that hypothesis for many years yet, but understanding just how extreme life can be on Earth shows us that, it is at least not impossible that life could exist in similar extreme conditions on other bodies in the solar system. And people who study that problem study extremophiles quite a bit. So that is what I'm going to leave you with. I've got 10:50 right now. So I'm going to stop recording. And once I'm done, we can cuss.